One of the things we said that has to happen to a DNA molecule before a cell can divide, and again, that's division either by mitosis or meiosis, is that the DNA has to replicate. Now, this was one of the challenges that Watson and Crick faced in creating the double helix model of the structure of DNA. A fundamental question was, how does the DNA replicate faithfully? Meaning, how is the information in the genes conserved or saved from cell division to cell division to cell division event? And Watson and Crick realized that the idea of complementarity between bases was the way that the DNA was to replicate faithfully. So let me explain to you a little bit more about what I mean on that. So here we have a double helix molecule. Again, we have the sugar phosphate backbones with the complementary nitrogenous bases paired together and held together by hydrogen bonds. So when the DNA replicates, one of the first things that has to happen is the double helix has to be denatured or broken apart. And then each of the original strands of the double helix will serve as a template for making a new complementary strand. So an enzyme, actually there are a variety of enzymes involved in this process, but the one that actually duplicates the DNA is called DNA polymerase. So when DNA polymerase sees an A on the template strand, it puts a T on the newly replicated strand. When it sees a C, it puts a G. When it sees a G, it puts a C, and so on. So you can tell what will happen is each template strand will produce a copy of the other template strand. In other words, a copy that is complementary to it. So that's what's being indicated. We have the dark shaded and the little lighter shaded strand. Each one of these will serve as a template for making a complementary strand. So this lighter strand that we see here will have the same sequence as the lighter shade gray strand that we see here. This form of replication is said to be semi-conservative because each of the original strands is preserved, but a new complementary strand is produced. And each of these complementary strands, again, are created by the idea of complementarity. A goes to T, G goes to C. Now, it's important to tie this back to the structure of the replicated chromosome. In other words, we said when a chromosome, which contains a single DNA double helix, replicates, you produce two sister chromatids, each one of them with a DNA double helix within it. And we said the reason the term sister was used was the two DNA double helices produced for the sister chromatids are identical. And now you can see how that works. This would be the DNA double helix in one of the sister chromatids. This would be the DNA double helix in the other sister chromatid, and both of these DNA double helices are going to be identical. So that when the cell divides by mitosis, each of the daughter cells gets a complete DNA double helix. And that's important because the double helix contains regions called genes, and the cell needs those genes in order to be able to make proteins and other products that it needs to function correctly. So again, this is the idea of semi-conservative DNA replication. The enzyme involved in the actual replication event is called DNA polymerase.